Welcome to a deep dive into two groundbreaking discoveries that fundamentally reshape our understanding of the adult human brain. First, we will explore the revolutionary finding that the adult human brain continues to generate new neurons throughout life, and second, how the brain distinguishes reality from imagination. Let us begin with the first discovery: adult neurogenesis, the birth of new neurons in the mature brain. For most of the 20th century, neuroscience held as dogma that the adult mammalian brain could not generate new neurons. Once development concluded, your neural complement was fixed. The old model suggested that neuronal birth occurred during development, then ceased entirely in adulthood, leaving us with a static neural architecture. In 1998, Eriksson and colleagues published a landmark paper demonstrating that humans generate new neurons in the hippocampus throughout adulthood using clever radiocarbon dating of DNA. However, the field erupted into controversy. Some researchers reported finding abundant neurogenesis, while others, using different techniques, found virtually none in adult human brains. The field split into two camps: those claiming adult neurogenesis was robust, and those arguing it was absent or negligible in humans. The resolution came from understanding methodological issues. The key problem was tissue preservation. Different autopsy protocols and tissue handling dramatically affected whether neural stem cells and immature neurons could be detected. Fresh, rapidly preserved tissue showed abundant markers of neurogenesis, while degraded tissue showed few or none, explaining the discrepancy. Between 2019 and 2023, multiple independent groups using optimized preservation methods provided definitive evidence that human hippocampal neurogenesis continues robustly into the 7th and 8th decades of life. The data show a gradual decline in neurogenic rate with age, but even in elderly individuals, hundreds of new neurons are generated daily in the hippocampus. Current estimates suggest approximately 700 new hippocampal neurons are born each day in healthy adults, with individual variation based on lifestyle factors. 700 neurons per day may seem modest, but integrated over years, this represents millions of new cells contributing to hippocampal circuits. This neurogenesis occurs specifically in the dentate gyrus, a subregion of the hippocampus critical for pattern separation and memory formation. The dentate gyrus, shown here in yellow within the larger hippocampal structure, harbors neural stem cells that continuously divide and differentiate into new granule neurons. The neurogenic niche contains a hierarchy of cell types: quiescent stem cells, rapidly dividing progenitors, and immature neurons gradually maturing over several weeks. This progression from stem cell to mature neuron takes approximately 6 to 8 weeks, with cells expressing different markers at each stage. But why does adult neurogenesis matter functionally? What computational advantage does the continuous addition of new neurons provide? The leading hypothesis is that young neurons, with their enhanced plasticity and excitability, improve pattern separation, the ability to distinguish similar experiences and memories. Young neurons help transform similar input patterns into distinct output representations, preventing interference between overlapping memories. Supporting this view, neurogenesis declines with aging and chronic stress, conditions also associated with impaired memory discrimination. Excitingly, we can enhance neurogenesis through behavioral interventions. Aerobic exercise, environmental enrichment, and learning new skills all increase neurogenic rates. Regular aerobic exercise can increase hippocampal neurogenesis by 50 to 100%, with corresponding cognitive benefits. We can model this as a fractional increase proportional to exercise intensity E, where 50% enhancement is typical. In summary, the evidence now conclusively demonstrates that human adult neurogenesis exists, is functionally significant, and can be modulated by experience. Now we turn to the second revolutionary discovery: how the brain distinguishes what is real from what is imagined. This addresses a fundamental question in neuroscience and philosophy. How does the brain determine whether a sensory experience originates externally or internally? The brain receives signals from both external sensory organs and internal memory systems. How does it tag them differently? Recent research identified a reality signal in the fusiform gyrus, a visual processing region that encodes not just what we see, but whether it feels real or imagined. The fusiform gyrus, traditionally known for face and object recognition, also contains populations of neurons that encode reality strength. Remarkably, this region activates both when we see something real and when we vividly imagine it. The key difference is in signal amplitude and pattern. Perception produces strong, consistent activation patterns, while imagery produces weaker, more variable patterns in the fusiform region. The second key region is the anterior insula, which acts as a reality threshold detector, monitoring fusiform activity and making the real versus imagined decision. The anterior insula receives input from fusiform gyrus and other sensory regions, integrating signals to determine whether activation crosses the reality threshold. Let me describe the elegant experiment that revealed this circuit. Subjects viewed visual noise, with faint stripe patterns embedded within it. The visual noise contained random pixel patterns with very faint oriented stripes that were near the threshold of visibility. While viewing this ambiguous stimulus, subjects were instructed to simultaneously imagine stripes oriented in a different direction. For example, real stripes might be vertical, while imagined stripes were diagonal, creating orthogonal representations. The key result was that occasionally, when subjects imagined very vividly, they reported perceiving their imagined stripes as if they were real external stimuli. This reality monitoring error occurred specifically when mental imagery was particularly vivid, and the real signal was weak or ambiguous. We can model this as the probability of mistake being proportional to the ratio of imagery strength to external signal plus noise. Functional MRI during these reality monitoring errors revealed the neural mechanism underlying the confusion. During normal perception, fusiform activity was strong, and insula correctly tagged it as real. During mistakes, imagery boosted fusiform activity above threshold, fooling the insula. Critical finding. When subjects imagined stripes vividly enough to mistake them as real, the fusiform activity pattern resembled actual perception of those stripes. The anterior insula, monitoring this fusiform activity, failed to distinguish the imagery-driven pattern from perception-driven pattern, allowing it to cross the reality threshold. This diagram shows the reality threshold as a dashed line. Green dots represent normal imagery safely below threshold, while red dots represent imagery that crosses threshold, triggering false reality perception. These findings have profound implications for understanding hallucinations in clinical conditions. Hallucinations in schizophrenia, Parkinson's disease, and other conditions may arise when the reality monitoring threshold becomes miscalibrated, set too low. With a lower threshold, shown in red, the same imagery strength that would normally be correctly identified as internal now crosses threshold and is experienced as real. Studies of schizophrenia patients during auditory hallucinations show increased activity in auditory cortex and fusiform regions with reduced insula-mediated reality monitoring. In Parkinson's disease, visual hallucinations correlate with attention network dysfunction, suggesting impaired top-down control over the reality threshold. We can model hallucination probability as a function of internal imagery strength, threshold setting, and attention control. An important question: can we quantify how much the threshold shifts in different clinical populations? Preliminary studies suggest healthy controls have high thresholds, prodromal individuals show intermediate thresholds, and active schizophrenia patients show significantly lowered thresholds. Let us perform a back-of-envelope calculation to estimate the magnitude of threshold shift required to produce hallucinations. Suppose in healthy individuals, the reality threshold is set at about 2.5 standard deviations above baseline imagery activity. This would give a probability of about 0.6% of spontaneously crossing threshold, matching the low rate of hallucinations in healthy people. Now suppose in schizophrenia, the threshold drops to 1.5 standard deviations above baseline. This would increase crossing probability to about 6.7%, a tenfold increase, matching the elevated hallucination rates observed clinically. Thus, a shift of just one standard deviation in threshold setting can explain the dramatic increase in hallucination frequency. These findings suggest therapeutic strategies aimed at recalibrating the reality monitoring threshold. Three main approaches are being explored: cognitive training to improve reality discrimination, real-time neurofeedback, and targeted brain stimulation. Cognitive training involves practicing reality discrimination tasks similar to the stripe experiment, potentially recalibrating threshold through learning. Training protocols typically span 8 weeks with progressive difficulty, gradually improving discrimination accuracy. Neurofeedback approaches use real-time functional MRI to show patients their own fusiform and insula activity, teaching them to modulate the circuit consciously. Patients see visual representations of their brain activity and learn strategies to raise or lower their reality threshold through mental techniques. Early pilot studies of neurofeedback training show 20 to 30% reductions in hallucination frequency after 10 to 15 sessions. Transcranial magnetic stimulation targeting the anterior insula represents a more direct intervention to modulate the reality monitoring circuit. Targeted TMS can transiently modulate activity in the insula, providing a potential therapeutic tool for recalibrating the reality monitoring circuit.